Welcome to the Astrobiology seminar. Some of you were able to attend the lecture last night. Well, before we get to today, next week, let's see, May 8th, we have Brad Dalton from NASA Ames Research Center, who will be talking about spectroscopy of Europa's surface in terms of the salts and possibly organics and so forth that are on the surface of Europa. So that'll be the Astrobiology seminar next week, and then there'll be a gap. It'll be 3 weeks after that will be the last of the year. Will be Joe Kirschvink from Caltech, and I don't know exactly what his title is, but he's always a fascinating speaker. But today's speaker, as I say, last night many of you heard Bob Hazen give a fascinating public lecture on an approach to the origin of life. Wide-ranging lecture, excellent questions and so forth. It was really quite an evening. And today we're delighted to have him give a technical lecture to the astrobiology community. But for those of you that were not there, let me just briefly say that he's a Renaissance kind of guy. He's a professional trumpeter. He's played with National Symphony, Boston Symphony. He's written 19 books on a wide variety of topics, most of them popular science books, but other books such as the history of brass bands in America, let's see, where's my other things here? The poetry of geology, why aren't black holes black, the diamond makers, et cetera, et cetera. He's also extremely interested in science literacy issues, and with Jim Treffel, he's written 2 books that are quite well known, Science Matters, Achieving Scientific Literacy, and a textbook called The Sciences, an Integrated Approach, which is used in introductory science courses where you want to have what are the key topics that should be known from all of the different areas of science. It's not just Astronomy 101, but you're looking for a student that wants to have a semester where they come away with key concepts in all of the sciences. So, we're delighted that he could be here today. And he's going to be talking about the origin of chirality, which he touched on last night, but we'll get into more of the technical details now. So take it away, Bob.
Thank you, Woody, and it's really fun to be able to talk about this aspect of our research at the Carnegie Institution on the origins of biochemical homochirality. And I want to start by just giving a list of some of our collaborators. We have so many wonderful people to work with. The thing that's striking to me about this list, and this will not be at all a surprise to you at the University of Washington, is not that there's so many people from so many institutions, but the range of disciplines, the backgrounds that these individuals have. We have people in organic chemistry, in microbiology, in ecology, in physics and chemistry, in molecular modeling, even a philosopher in this list. So the idea of having research collaborators from many different fields and coming together to try to tackle these questions is so critical. And I certainly want to thank them for their contributions, and many of the things I'll talk about today come from the work of these individuals. I want to look at 2 possibly related questions today. The first has to do with just the interaction of crystals and organic molecules. And I think that's a very general and broad question. But we're also going to look specifically in the origin of life and what process the early Earth geochemical environments might have selected and concentrated certain molecules out of a prebiotic soup. So that particular idea. This idea of crystal molecule interactions that has tremendous applications to a whole wide range of disciplines. And some of them may be a sort of a surprise to you, but just think about molecules and crystals. Teeth and bones, they're composite materials formed of proteins and for example, hydroxyapatite. When you think about the process of biomineralization, the way living things secrete minerals, the formation of shells, for example, or some microbes that do biomineralization, formation of biofilms as well. Fossilization, the transformation of a living thing into a rock, involves all sorts of mineral molecule interactions. Weathering and soil formation, critically important, and indeed I've read some articles recently that indicate that microbial action accelerates weathering in many rocks by orders of magnitude. So this is certainly a very important kind of interaction. Very practical technologies, paints, glues, dyes. Anytime you have an organic material sticking to an inorganic material, and as well as lubricants. When you think about it, that's another kind of mineral-molecule interaction in many situations. Environmental monitoring, cleanup, turns out understanding the interaction of organic materials and crystals can be very important in this regard. Nanotechnologies, and this certainly will be an important future area to understand. I'll talk later today and allude to drug synthesis and purification, another place where this becomes very important, and I will argue also the origin of life. All these fields and more are places where a fundamental understanding of how crystalline materials on the one hand and organic molecules on the other hand come together, how they interact at the molecular level, is fundamental to our understanding. So, let me give you just one fascinating example of how this can come into play, and also how this idea can lead you in new directions you never would have expected. This is a tiny portion of a bone. And what you're seeing here in purple are 5 atoms of calcium that are part of a hydroxyapatite crystal in the bone. Bonded to this hydroxyapatite are 2 small parts of larger molecules called osteocalcin, proteins that add some of the strength and the characteristic properties to bone. So here you see 2 glutamic acid residues, 2 from a second osteocalcin molecule, and you see how in this work by Hong et al., which was published a few years ago in Nature, there's a very strong interaction between the mineral and the molecule, an intimate interaction between glutamic acid and those calcium atoms. Well, that's fascinating, but it leads to some consequences about osteocalcin in the fossil record. It turns out that because of this strong interaction, the protein osteocalcin can survive for tens of thousands of years in a fossil bone, whereas if it were just an isolated protein, it would denature very quickly, it would break down, you'd never see it. But because it survives in fossil bones, you can take fossils of bison or ox or other animals, you can extract the osteocalcin, you can sequence it, and actually get phylogenetic information. And what people are now doing is creating phylogenetic trees for extinct mammals, and they're doing it by sequencing the proteins that are preserved in those fossil bones. You may have seen recent articles about the preservation of collagen in much more ancient dinosaur bones. I think that's the kind of thing. This would not be possible were it not for the strong interaction of the molecules with the mineral substrate, which protects, preserves, and prevents the degradation of the bone. So that's just one example. We are learning things about the evolution of macrofauna from the consequence of interactions in minerals and molecules. Now, today I'm going to talk about the origin of life. And just to review, kind of give you an echo of last night's lecture, I make a few basic assumptions when I talk about origin of life. The first is that the original life forms were based on organic molecules. They're carbon-based life forms. The raw materials, oceans, atmospheres, rocks and minerals, those are your building blocks. And that the origin of life required, as I talked about in detail last night, a sequence of emergent steps, where you go from simplicity of a geochemical world to the complexity of a biochemical world through a series of chemical complexifications. In specific, the 4 steps that I talked about last night. First, the emergence of the simple biomolecules, the building blocks, the amino acids, the sugars, the lipids, the bases of DNA and RNA, and so forth. The second step, which I'm going to focus on today, is this emergence of organized molecular systems and the selection and concentration of a few molecules from many. The third step, you'll recall, was the emergence of self-replication, molecules that make copies of themselves in a system, and finally the emergence of natural selection. Those 4 steps are key, but we're going to be looking at this second problem. The origin of biomolecules, the problem. Fundamental aspect of life is a high degree of molecular selectivity. If you go into the databases for E. coli, for example, you'll find that there are only a few hundred different small molecules in E. coli, out of all the millions of small molecules that are in Bielstein. Prebiotic synthesis is basically indiscriminate. You make hundreds of thousands, millions, perhaps more, small molecules. Life uses only a few. So the question becomes, what prebiotic processes could have selected and organized that vast array of organic molecules to give you just the right ones that are organized, that are synthesized down?
What's your definition of small?
Small molecules, 8, 10, 12 carbon atoms maximum. So we're talking about things, really monomers, if you will, in the biological scene. let's look at amino acids just as an example of what I'm talking about. If you look at Murchison meteorite, there are over 90 different amino acids so far identified, compared to the only 20 in biological systems. Those amino acids in biological systems are restricted to alpha hydrogen amino acids, as opposed to Murchison, which, for example, has a great many alpha methyl amino acids. And finally, this question of homochirality, the handedness, that in living systems, overwhelmingly you have left-handed amino acids, whereas in Murchison and in other natural products, there is a more equal ratio. Now, Murchison is interesting that it seems to have a slight excess of some L amino acids, and we can talk about that later on. But the basic point is that in an early Earth, where the amino acids would have had a long time to sit in an environment where they can flip back and forth, you predominantly would have had a 50-50 mixture of amino acids from which to select, and yet life is predominantly left-handed. How do you get this selectivity? Here's the problem: homochirality. Life's molecules come in left and right-handed forms. The prebiotic processes produce 50-50 mixtures, and yet life is largely homochiral. That's the question we're asking. How did this happen? And even if I can't give you an answer, and even if you're not convinced by what I say today in terms of homochirality, as I alluded to last night, the chiral drug industry is huge, $200 billion a year. And it's because to purify left and right-handed molecules is very important for medicinal purposes, for biological purposes. Our bodies respond differently to left and right-handed molecules and left and right-handed drugs. Let me give you some basic vocabulary, because I slip into these words very easily and they may be unfamiliar to you. So just to give us a common ground, the word chiral is synonymous with enantiomeric, or handed. So we're talking about a chiral molecule, I'm talking about a left or right-handed molecule, for example, mirror image pairs of molecules. The designation for a so-called right-handed molecule, which is a rather arbitrary definition in any case, sometimes they're called D, dextrose, sometimes they're called R, and you'll see those interchangeable. Similar for left-handed molecules, sometimes you see L and sometimes you see S. The word sinister is related to that same root for S. Homochiral means that you have all the same handedness in a collection of molecules. Heterochiral is a mixture of handedness. Racemic is the word for a 50-50 mixture. So if I talk about racemic molecules, it means that there are equal numbers of left and right-handed molecules in the mix. And symmetry breaking. Symmetry breaking is the process by which you take a racemic mixture and separate out the left and the right from each other. You break the symmetry of this collection of molecules. So those are words that I'll be referring to throughout the talk. Let's look at why chiral purity is important. Here's this case of limonene, the flavoring where the right-handed form smells like oranges and is used in that context, but the left-handed form smells like lemons. And it's because the taste receptors in our mouth are shaped such that they're handed, and a left and right-handed molecule will fit differently into that receptor. And I mentioned thalidomide last night, this tragic case where the right-handed form was given to women to cure morning sickness, but the left-handed form caused birth defects. And indeed, even if you took a chirally pure form of thalidomide, in your body it racemizes, it flips from left to right very quickly, so that the right-handed form in part becomes left-handed form while it's in your body, and therefore there's no way of taking thalidomide safely. And only recently has come back on the market for a very limited use in treating certain kinds of cancers. It turns out to be very, very effective for a cancer called multiple myeloma. But my mother, who had that disease and was given thalidomide towards the end of her life, she was 80 years old and she had to sign a sworn statement that she would not get pregnant for legal reasons. There's some very sad irony in that, I thought. But that's the story. Enantioselective chemistry. The problem facing the drug industry is the same problem facing early life. How do you start with a racemic mixture and get a pure product? And there are two ways to do this. The first is an enantioselective separation, where you start with a 50-50 mixture, synthesized by some normal process, and then you pass it over a column or some kind of environment that's chiral, which separates out, typically by time, the left and the right-handed molecules. So you collect the right-handed molecules, then you collect the left molecules through a chromatographic technique of some sort. The other equally important form is called enantioselective synthesis, where you start with reactants that are themselves not chiral. They're not handed, but they have the potential, if you add just one more component to them, they will become handed. And if you have a chiral catalyst, it turns out that you can take some prochiral reactants, move them over the chiral catalyst, and the product will be chirally pure. So that's another way of doing it. And actually, each of these possibilities may have obtained in a prebiotic setting as well.
That one component, there'd be two ways to add it.
Two ways of add it, and it just basically, if you add it over here, and it has to do with if you have a carbon atom that has three things bonded to it, and you add a fourth, you can either add it to this side or to this side.
What's a way to distinguish? I mean, chemically, how does one do that?
You use a particular chiral environment that happens to bond to the prochiral reactant, and it can only stick on the other component from one side. So you have, for example, a hydrogen atom over here, you have an amino acid amino group over here, you want to add on a phenyl group. And the phenyl group is sitting on the surface like this, and this is the only way that the three-bonded thing comes in there. A double bond becomes a single bond. You now have a four-bonded carbon, which is a chiral center. So there's ways. And engineers can actually figure this out in great detail. But in a prebiotic sense, we have to sort of start from scratch and see what might have happened. So here's the problem again, just in a nutshell. Prebiotic synthesis processes give you mixtures of left and right. Life demonstrates this chiral selectivity. What is the mechanism of symmetry breaking? And there's a huge literature on this. It goes back almost 100 years. And there are many different kinds of hypotheses. Some of them have to do with global mechanisms or universal mechanisms. The idea that the left-handed amino acids that we see on Earth were predetermined, that this is a deterministic aspect of life. In some cases, it has to do with the deterministic aspect of our region of the galaxy. For example, if there's a rapidly rotating neutron star producing circularly polarized radiation, that could selectively photolyze right-handed amino acids so that only left-handed amino acids survive. And presumably there are other regions of the galaxy where the opposite photolysis would have occurred. But we happen to be in a region where you get this preponderance of left-handed amino acids in the nebular milieu. Now, another example, and this is discussed by physicists, this is the one symmetry, universal symmetry breaking aspect. And basically you can think of it, when you have beta decay, an electron is released, and that electron is circularly polarized, and it always spirals in one direction. So there is a slight chiral bias when that electron hits another molecule, or if it gets involved in a chemical reaction, it will bias that chemical reaction slightly. Very, very small energies involved here. I am unconvinced by this mechanism, but there are some physicists that will point to this as the one sort of handedness of the one thing that would not be mirror symmetry if you looked at a mirror, the through the looking glass analogy, that the other side of the mirror universe looks identical to ours except for this one physical attribute, the handedness of the rotation of beta decay. Those are global or universal mechanisms. But they're also local chiral microenvironments. And if you will accept the idea that the origin of life was a chemical event that occurred in a microenvironment, at some point the juxtaposition of just the right molecules and maybe just the right surfaces, then you have to think about, are there local environments that are chirally biased? And indeed there are very strong local chiral environments. One of them is the chiral molecule itself. I think a very plausible idea, one I'm not going to talk about today, but a very plausible idea is that a chiral molecule itself attracts chiral similar molecules, and that so for perhaps a chain of peptide chain, a long chain of amino acids, maybe it forms preferentially with all L and all D molecules as opposed to a mix-up of L's and D's. Maybe that just doesn't bond as easily or form as easily. Or maybe there's a selective hydrolysis that LL and DD bonds in amino acids are stronger and last longer than a DL bond, which, if it's selectively hydrolyzed, ultimately you winnow those out and you get homochiral sequences. That's possible. And people have speculated on that. There's not a whole lot of experimental work to show one way or another, and I'm not going to discuss that possibility, but it certainly is out there. Another option, another very strong chiral environment, which I will talk about today, are mineral surfaces. So, to give you my prejudice here, It's not that minerals are the answer to the problem of life's homochirality. It's that minerals provide very strong local chiral environments, and they also have the ability to select and organize molecules on their surface in a way that could lead to higher levels of organization. And so I think we have to look at these environments just as an intrinsically interesting property, something that might have very important commercial or industrial applications, and also one that to me is as good a possibility as any for the origin of life's homochirality. That's sort of where I'm coming from. So the hypothesis is that minerals work in this regard, that left and right-handed faces of minerals, if you will selectively concentrate left and right-handed molecules. And what I want to do is sort of go through this. I will tell you briefly that our recent work shows that, in fact, this is true. We can demonstrate this experimentally, and I'll give you some more insight on that. These are just some of the selective left and right-handed molecules. The TCA is a tricarboxylic acid on calcites, on feldspars, on quartz. We're talking about the commonest of rock-forming minerals here, things that appear in all sorts of geological environments. Today I'm going to do 4 things. So the structure of the talk, I'm going to first examine the occurrence of chiral mineral surfaces in nature. How common are they? What is the likelihood that in a geochemical environment you're going to find a chiral surface? I'm then going to look at this process, chiral selectivity, and demonstrate experimentally that, in fact, selection occurs. Left and right-handed molecules can be separated by minerals. Third is to look through a theoretical analysis, first principles calculations, of exactly what's happening at the molecular scale. Why would one molecule stick to one particular surface preferentially? And finally, I'm going to propose a more general way of approaching this problem using a combinatoric technique based on DNA microarray technology, where we can study literally thousands of mineral molecule pairs in a single experiment, which really makes our life a lot easier. Okay. So first, natural chiral surfaces. Now, natural chiral surfaces occur in sort of 3 variants. I'm only going to be talking today about the first one, but that's the least likely to occur in a natural environment, and I admit that up front. That's just a crystal termination that sort of idealizes a set of almost planar of atoms coming to a surface, on a flat surface. Much more likely you have stepped surfaces because of growth defects, or even more likely kinked surfaces where you have those kink sites are chiral in nature. The very essence of a site like this is a perfect place for a small molecule to nestle in an environment that's handed. And this we see over and over again, even in metal surfaces. Now, let's just look at one of these kink surfaces. It turns out that the chemical engineers, they primarily study things that are close-packed cubic structures like platinum or gold or copper. And the reason is those are highly catalytic, and they can also prepare them quite easily. But you say, but gee, close-packed surfaces, why would they be chiral? They're chiral because they have all these kink sites. If you have a high-angle surface, like the 6-4-3 surface of a metal, then you have the left-handed face, which is non-superimposable in terms of its kink structure in the right-handed face. And so you have all these little nooks and crannies where either left or right-handed molecules fit. And for the last decade or so, people have modeled, using theoretical approaches, small molecules nestling into these kink sites, and have shown how you can get a selective absorption. And typically 0.5 kilocalorie per mole is the preferential energy of a leftover right-handed molecule on this kind of surface. Here's another calculation of what might happen. Experiments are much, much more difficult in this case, but calculations at least indicate the theoretical possibility.
Basil, what is the difference between the 2 surfaces, left or right?
What we're seeing here is just whether the kink sites basically come in from the left or the right. So if you have a crystal of a metal and you polish one surface, its kink sites are going to be facing this way. And if you polish the opposite surface, the kink sites are facing the opposite direction. They basically are mirror symmetry forms. And as I showed in this figure you go to back, look at the black versus the red kink structure. Those 2 kinks represent the left and the right-handed face, can't superimpose on each other, just like your 2 hands can't superimpose. So that's what we're talking about when we say left and a right-handed face. Presumably, if there's any selection, right-handed molecules would go on one side, left-handed molecules on the other side. And that's what the calculations show, with a preference of about 0.5 kilocalorie per mole. That's typical for small molecules on these kinked surfaces.
What fundamentally makes it not be just a regular corrugation?
It's just because you have an offset of 1 versus 2. There are 3 different kinds of contacts between a close-packed sphere and its neighbors. And if you have one kind of contact this way, a second kind of contact this way, and a third kind of contact this way, that gives you a handed thing where the two halves are mirror images. So the kink sites basically are mirror image pairs, just like holding your three fingers this way as a mirror. That's how they nestle in. And it's not intuitively obvious. I mean, in fact, no mineralogist even thought about minerals in this way prior to the work we've been doing. So, except for the case of quartz. Quartz is the one common rock-forming mineral that occurs in left and right-handed forms. And the crystals actually show little forms where these are called window faces, and if this little face goes up to the right, it's a right-handed crystal. If this little face goes up to the left, it's a left-handed crystal. And the difference structurally is that they're SiO4 silicon tetrahedra that basically are connected in helices, and the helix can go to the left or it can go to the right. So you have left and right-handed quartz crystals. I've got an example here. This happens to be a left-handed crystal, and these grow in profusion, and you can find 50% left-handed crystals and 50% right-handed crystals. I'll pass this around so you have a chance just to look more closely at the kinds of crystals. And for almost 100 years, people have used these, and tragically what they do is they take the crystal and they powder them. They crush them to a fine, fine powder, so you're increasing the surface area of your left or right-handed crystal. Sounds good in principle, I suppose, but it's a terrible, terrible thing to do. Not only does it destroy beautiful crystals, which I hate to see as a mineralogist, but also you're destroying all the information that you might get from the surface. Here's this specimen of quartz I collected when I was a teenager, Paterson, New Jersey. I was a mineral collector, and I found this fascinating crystal in which there are very faint coatings of iron oxide on different quartz faces, but if you look closely, you'll see that some faces are coated and some faces are not. Quartz is a trigonal mineral. It has threefold symmetry, and the 101 faces are coated, the 011 faces are not, indicating that different faces have different surface structures, different absorption processes. In terms of molecules, it's just as likely that the left-handed molecule will be absorbed in one face and a right-handed molecule in another face, because they're completely different surface structures. Nothing intrinsically left-handed or right-handed about the faces. It's just an arrangement of atoms. And so you have to look at it face by face. You crush the crystal. You destroy all the information. So we don't do that. We look at individual crystal faces and try to characterize those faces as carefully as we can. Here's a wonderful drawing by Steve Parker, which sort of shows a quartz crystal with its different surface structures. You can see that every face has a different termination with yellow oxygens and red silicons. I've been doing this as well. Here we have oxygen shown in red. The X's mark the atoms that are closest to the surface, and you'll see that here's the termination of a quartz 100 face. That's the prismatic faces of the quartz. And very interestingly, those are not uniform heights. Where these oxygens intersect the surface, you have quite a bit of topography. So this is kind of an interesting surface. It's chiral, and it also has docking sites and those sort of grooves and wedges on the surface. But look at the 101 face. This is the one that absorbs iron oxide. It's a chiral mineral, but the surface manifestation of the atoms is very flat. Furthermore, there's a mirror that runs right down the center of that. So this is not a chiral face. Even though it's a chiral mineral, there's no point in studying the 101 face with chiral molecules. So it doesn't get you anything. And here's the 011 face, and it's got some surface irregularities, and the distribution of surface oxygen also is slightly chiral. But that's quartz. Quartz turns out not to be a very interesting mineral in terms of its ability to separate left and right-handed molecules. Feldspar, however, is much more interesting. Feldspar is a very, very common mineral. Here's a crystal that has these left and right-handed faces, a mirror plane running right down the center here. Gives you two faces with mirror image, and perhaps left and right-handed molecules could be separated on a face like this. When we look at that quartz crystal structure, you see that there's quite a bit of surface topography, and furthermore, some of these blue atoms, those are an alkali atom that could be a potassium or a sodium, or a calcium in the case of a plagioclase feldspar. You get both positive and negative bonding centers close to the surface. So there's a much more interesting bonding environment in feldspar. So this is something we want to look closely at. Diopside, the commonest mineral, magnesium and iron. has also some surface irregularity and also has both positive calciums or magnesiums and negative oxygen atoms near the surface. Quite a bit of topography. Again, very interesting bonding environments for organic molecules. And my favorite, the mineral that is so important in terms of biomineralization, forming shells of clams and snails and echinoderms and all sorts of things, calcite. And calcite, the commonest crystal form, this 2-1-4 phase that you see here. Typically, you'll get a crystal that comes to a point like this, and there are 6 of these faces that form coming to that point. 3 of those faces are left-handed, 3 of them are right-handed, and the surface structure is very strongly chiral. That is, it deviates strongly from any mirror symmetry. You have both positive calcium atoms, negative oxygen atoms near the surface. You also have a very strong topography with grooves that are about 2 angstroms deep running along the surface of those crystals. And this makes incredible docking sites for organic molecules. We have positive and negative charges distributed in a very asymmetric, non-mirror symmetry way, plus grooves in which to dock things. So calcite becomes extremely interesting. Now, I can sort of describe these in a qualitative sense, but it's much more interesting to be able to quantify the degree to which a surface might be able to select chiral molecules. And so we have developed what's called a chiral index, an index which basically says when you have a surface structure and you fold it on top of itself, how closely do they fit together? Do they match precisely, in which case the chiral index is 0? Do they mismatch a lot, in which case the chiral index gets larger? If you have a mirror symmetry, as in the calcite 104 surface you see here, the chiral index is 0 because there is a mirror symmetry built in. But if you have a very non-mirror symmetric sort of surface, like the calcite 214, the surfaces don't match at all when you fold it over, and so you have a high chiral index. And so you can quantify these. These are all chiral indices. They're given in angstroms. It has to do with how closely atoms, when you fold things over, match with each other. And the average displacement tells you in angstroms the average distance between the mirror image pairs. The maximum displacement tells you the maximum displacement of an atom when you fold it over on itself. You see quartz here. It's 0.4, 0.5, not a very high chiral index. Copper, the metal, is much, much better. That's what the chemical engineers use. So they see an average displacement of 0.84 angstroms and a maximum displacement of almost 1.3. But look at calcite. This is the highest chiral index of any material we've seen, much higher than that of metals, and makes this an extremely interesting material, not only for origin of life studies and just other basic studies in chirality, but also for industrial processes. That if you can get a surface like calcite to select the molecule that you want, you're going to have a very good selection indeed. So, basically the conclusions, there are lots of chiral mineral surfaces in nature. Indeed, any rock that you see on the ocean floor, anytime you split open a rock, it's just loaded. The chiral surfaces are all over it. So chiral surfaces are everywhere. Yes?
Can you quantify it? Given that different surfaces of the same crystal have different chirality indices, what fraction of the surface area is like that one face? It could be a tiny little face that's really chiral.
It's really going to depend on the rock and the rock's fabric, but in many cases, you're going to find that more than half of the surface that's exposed will be chiral. In fact, the majority of surfaces, unless you have a crystal growth face that happens to have a mirror symmetry running right down to it, the majority of growth surfaces and the majority of fracture surfaces are going to be chiral. So if you have a porous rock, you're talking about huge areas of chiral surfaces on which you can do absorption experiments. This is a really significant part of the natural environment. It's interesting to me that the intrinsically chiral surfaces of quartz possess relatively low chiral indices and make quartz less interesting than some other minerals, whereas in oxides and silicates you see much, much larger chiral indices if you have both positive and negatively charged atoms near the surface, as you do in calcite, as you do in diopside, as you do in feldspar. Also, the relatively large chiral indices are often associated with stepped and kinked structures, as we see in the metals. But this also means that in the real world, where you have natural growth features on the mineral, natural minerals are not perfectly flat. They have hillocks, they have etch pits, they have all these sort of surface topographies, and those may be far more important in this process even than the idealized surfaces that I'm going to be talking about for much of today's talk. So keep that in mind, that we recognize there's all kinds of chiral environments, microenvironments of mineral surfaces. Now I want to talk about briefly the experiments I've done on chiral mineral selection. And this was done with my dear friend Glenn Goodfriend. He was a lifelong collaborator with Steve Gould and many other notable people. Both Glenn and Steve died about 3 years ago. And we were in the middle of these experiments, and it was really a tragic loss for science for both these people. But Glenn in particular, he was a master at analyzing left and right-handed amino acid mixtures and getting precisions and accuracies in his measurements that no one else I've ever seen has been able to match. Truly astonishing. I give him blanks and all sorts of sort of test cases, just slip them into my runs, and it was amazing how he was just zeroed right in all his experiments. He was just a master at this analysis. What we did is we took calcite crystals, much like the one that's passing around. Took the crystal, dump it into a beaker that has a 50-50 mixture of left and right-handed aspartic acid. That's an amino acid that is known to absorb strongly onto calcite. We let it sit overnight. We pull the crystal out. We wash it off. We then have 6 faces coming to a point. 3 are left-handed, 3 are right-handed. Meticulously take a pipette. This is all done in clean rubes with total body covering, because the slightest contamination will skew our results. Carefully put a little bit of hydrochloric acid on each surface, pipette it off, put it in a vial, and then one by one analyze those surfaces. Repeat that 3 times on multiple crystals over and over again to make sure we're not somehow fooling ourselves that we're seeing an effect that we hope to see. Here's one of Glenn's GC analyses. What you see here is the baseline and the aspartic acid doublet. Blowing that up, you see how clean his baseline was. You see the separation about 15 seconds for the left and the right-handed aspartic acid. And it's the area ratio of those 2 peaks that tells you the D to L ratio of left-right-handed amino acids. Here are the results of one set of experiments. And what you see here, this is kind of an unusual graph. You see 6 vertical columns representing 6 faces coming to a point on the crystal. 3 of them are right-handed. The other 3 are left-handed. We measure the D to L ratio. The solution itself is very close to 1, a 50-50 mixture. So this would be excess L, that's excess D. The key here is we start with a 50-50 mixture. Had we seen an L excess on a few crystals, it would mean absolutely nothing, because the entire environment of our laboratory, of our lives, is contaminated with L amino acids. What we need to see is an equal and opposite separation of D excess on one set of faces and L excess on the other set of faces. Without that equal opposite separation, there is no result here. Furthermore, we ran many tests on cleavage planes which have no chiral bias, and those all better come out very close to that horizontal line, or again, we're fooling ourselves. So what we found systematically over and over again is a slight excess of D amino acids on the right-handed faces, a slight excess of L amino acids on the left-handed faces. And this paper, which was published in PNAS some years ago, is the first experimental demonstration of the separation of left and right-handed molecules on a crystalline face. We've subsequently done this on a number of other minerals and molecule pairs. But the conclusion is that we can separate left and right-handed molecules. We see the separation of aspartic acid on calcite, but we do not see a similar separation of either glutamic acid or alanine on calcite. So the question is, why do we see one and not the other? The maximum absorption also occurs on faces that have terraces or steps. And it makes us wonder whether the effect we're seeing might not be amplified by step edges where these molecules may be nestling in along a specific linear feature, which is really interesting because, of course, if you align amino acids and then can bond them together, then you form a peptide chain, and the chain's going to be homochiral, all the same chirality. So that's a very interesting possibility. Okay, so now the third step is to model these not experimentally, but theoretically, trying to understand how the molecules are nestling in on the surface. Why would you get left-handed aspartic acids bonding to a face but not right-handed aspartic acid? And that's what we wanted to find out. So we're basically going to use density functional theory. Experiments can't really help us about the details of the molecular interactions. And this kind of modeling, then, is what I'll be describing in this section. This is work done with Aravind Astagiri, who is a graduate student at Carnegie Mellon in the chemical engineering department, then came to work with me as a postdoc, and now is a professor at the University of Florida. What you do is you start with a model. This is a model that's generated by computer in which you have an alanine molecule, for example, floating above a calcite surface. It's an idealized calcite surface. And with density functional theory, you model basically the behavior of all the electrons in this system, except for the core electrons, which are treated by a pseudopotential. So basically, you allow the whole system then to minimize its energy. It's a very time-consuming process. It involves a lot of computer code. Aravind's an expert in this, and this is what he found. So we use this first principle method. By the way, we're ignoring water in this particular experiment, which one can argue actually makes the modeling not necessarily valid for our aqueous solutions. But what we find over and over again is that the amino acids are in fact bonding primarily to the surface, not through any intermediate hydroxides or hydrogens. So we think that this does give us a valid picture of what's going on. And we have to look at numerous plausible configurations. You have to start with the molecule in all sorts of orientations above the crystal surface in all different places. And so we sort of have to have an intuition of what might be a plausible starting point, and then you minimize those. So we looked at dozens and dozens of plausible configurations and really quickly zeroed in on what were the logical bonding sites for an amino acid on a calcite surface. And we find that all the stable configurations involve forming strong calcium-oxygen bonds, calcium of the calcite to an oxygen in the amino acid. Okay, so you begin by taking a D-Alanine molecule, bring it to an unrelaxed surface, and then you find in the final configuration that you're forming two strong bonds, one an oxygen-hydrogen bond, that's from here to here, the other a calcium-oxygen bond from here to here. And this is how alanine bonds to the surface. Notice also the relaxation of the calcite surface. Here's, for example, a CO3 group. You see how it rotates. See also that the calcium atoms, which are aligned here, become staggered over here. This all has to do with the interaction of the molecule and the surface. So
it rotates around just to accommodate the size of the
this is basically
of the aspartic acid?
Yes, and if you go down 2 unit cells, there's no distortion of the calcite to speak of. It's only right at the surface that when the molecule comes in, it causes this surface distortion rotation right at the surface area. So this is the kind of interaction which has been well documented in other materials. This is not at all surprising, but here is a particular docking site of alanine onto a calcite 2,1,4 surface. And here is why you don't get chiral selection. Here's a D-alanine and an L-alanine bonded to this 2,1,4 surface. 2 bonds for the D-alanine, 2 bonds for the L-alanine. Essentially identical configurations. And this is just as if you took your thumb and forefinger and tried to touch a surface. They're identical. The energies are identical. There's no difference. And you could pick up an object with these 2 fingers and these fingers equally well. So there's no preference of a left or right-handed molecule here. Now look at aspartic acid. Totally different situation. The D-aspartic acid bonds to that calcite surface with 3 strong bonds. And indeed, 2 of them are calcium-oxygen bonds because of a fortuitous spacing between this oxygen and this oxygen compared to the 2 adjacent calciums. And there's the 3rd bond. But if you have a left-handed aspartic acid, only 2 strong bonds form. And here's the key to chiral selection. Chiral selection is like the classic problem of a bowling ball. If you go bowling, you can't, if you're right-handed, you can't use the left-handed bowling ball because there are 3 holes and they're not aligned, they're non-collinear. And so this is a right hand, this is a left hand. It turns out just purely by fortuitous coincidence, you have 3 strong charge centers on the aspartic acid that match 3 charge centers on the surface of the calcite 2,1,4 surface. So the D-aspartic acid sticks really strongly to the surface, the L-aspartic acid not so much. And here we have an 8 kilocalorie per mole difference in bonding energy. So in terms of these particular sites at room temperature, that would correspond to a 99.9% preferential selection of one-handedness over the other. We don't see that because of their other experimental effects that come into play with solutions and small amounts of solution being left over and so forth. But in terms of chiral selectivity, this is by far the strongest effect ever seen, certainly in a calculation. And we think we can understand in detail, first of all, why alanine doesn't show chiral selection, why aspartic does, and why this is a very strong effect. So the conclusion is that we can model the chiral interaction. It requires 3 strong points of interaction that are non-collinear. We can see which molecule sticks to which surface, but it's idiosyncratic. It has to do just with the fortuitous distribution of charge centers on the surface. And now we're going to look at a general research strategy, because I'll tell you, life is too short to take calcite crystals and dump them into aspartic acid, and then dump them into alanine, and then dump them into glutamic acid. Each of these experiments takes weeks or months. The analysis of each individual sample is very meticulous. We needed to find a simpler way. And so what we're going to do is a combinatoric approach using microarray technology. My colleagues at Carnegie who have helped me with this, Jake Mall, Andrew Steele, Rebecca Martin, all of whom have training in the biological sciences, and here's a great case where interdisciplinary research has paid off. Chip writers are devices that have been typically used to study different DNA sequences or proteins and their interaction, their reactivity, and so forth. This is a very important technology that's coming along in medical diagnosis, and you will soon be able to go to your doctor and he will have chips that contain millions upon millions of targeted DNA sequences that, for example, relate to specific genetic diseases, and see whether you've got them or whether you don't. What we can do is we can take up to 126 1 centimeter square chips of minerals. On each of those minerals we can put up to 49,000 plus spots, tiny spots, 100 microns in diameter, array them on the surface, drawing from up to 96 different wells. So one could have left-handed aspartic acid, one could have right-handed aspartic acid, and all the other amino acids and the pentose sugars and the carboxylic acids and whatever else you want. Lots of different small molecules. And on each of these different mineral chips, you can array a series of spots, varying in concentration, perhaps mixing different molecules together to see selective absorption of one over another. And the traditional way of analyzing these spots is to use fluorescent tags. So our first experiments were just to show that we could array spots, 100 micron spots, onto a flat mineral surface of things like amino acids, which had never been done in this way before. So here is an array of basically fluorescently labeled amino acids on glass slides to show that we can make arrays of concentration series. The traditional way of analyzing microarrays is through fluorescence, where you basically just look at fluorescent markers and see which spots shine up and which ones don't. And we certainly started with that idea. This is quartz, a left-handed and right-handed quartz, looking at a fluorescently labeled L-lysine. And what you'll notice is that the L-lysine sticks much more strongly to the right-handed surface than the left-handed surface in these experiments, which is interesting, which is fun, and which is absolutely irrelevant to the origin of life, because even though we can print these microarrays, these are fluorescent tags that are themselves big molecules, sort of stuck onto this small molecule. And so what you're seeing sticking on the quartz surface is not left or right-handed lysine, it's left or right-handed lysine with this great big thing sticking on the side, and that's really what's interacting with the surface. So proof of concept is great, but it doesn't relate to origin of life at all. So we have to figure out a way to do this without fluorescent tags. Here is lysine without a fluorescent tag, arrayed onto calcite. And here you see spots that are 100 to 150 microns in diameter. You see some of them are sort of irregular, some of them are nice and round. So this is reflected light, just showing where we've done these arrays. And this is work done with Ed Vincenzi and Detlef Ross at the Smithsonian Institution, where they have a ToF-SIMS. ToF-SIMS is time-of-flight secondary ion mass spectrometry. It basically means you take a beam of ions, you direct it at the surface, the ions will strip off and fragment any molecules adsorbed to the surface, and then you collect in a mass spectrometer the characteristic fragments. So if there's an amino acid, you'll see characteristic CHN fragments. If there's a sugar on the surface, you'll see characteristic CHO fragments, and so forth. And you can look for those and basically analyze what molecules may or may not have stuck to a surface. Here, for example, are ToF-SIMS results on a single 150 micron diameter spot of lysine, L-lysine, on a calcite 214 surface. That's that chiral surface. And what you see here are various fragments. Most of them that are showing as bright spots are CHN fragments of various sorts. You also see masked, this is a calcium spot. So the spot masks the calcite. And so here's a very simple way of telling whether something has stuck or not stuck to the surface. You basically do a spectrum on the spot, you do a spectrum off the spot, you do a difference, and basically by looking here in red is just the calcite, you'll see very strong mass fragments related to the calcite. You also see very strong fragments related to the amino acid. And you can do a difference and therefore come up with a quantitative measure of how much lysine has stuck to that surface. So this is a nice technique. We still had some problems here. We had very dirty surfaces, but we've finally figured out how to take care of that with plasma cleaning of these surfaces. We used this high resolution detail. And we had some additional problems which we basically resolved now by cutting very precise 1 centimeter square, 0.3 centimeters thick slices in the minerals, right off crystals like the ones that I've been handing around. Just cut them out very carefully, and a gem cutter does this to give us the best possible structures. And then we can print an array. So here's a map for an array. In this particular one, we have 4 different pentose sugars, including ribose and the 3 other pentose sugars, and also D-Lysine, L-lysine, and DL-Lysine. So this was another sort of test, a trial run to see if we could both print the array. There's what the array looks like in reflected light before the crystal is washed. So you print the array, you then wash the crystal, and you see what sticks. And here's that particular Feldspar crystal. It's in a new special holder we've made that gives you a very precise leveling and also allows you to get back through X, Y coordinates exactly and refine your spots. Because if you washed off a spot, it's hard to know where it is. So you want to find out if there's anything preserved there. And then you can do this kind of mapping. So here are spots of D-Lysine on Feldspar on this particular surface. Here's a mixture of D and L-Xylose on Feldspar. That's one of the pentose sugars. And again, you can do difference maps. You can map the spectrum. Here's the mass fragment right around 40, mass 43. What you'll notice is that there's a characteristic CHN peak here of amino acid, which is not present in the sugar. Very strong peak for the sugars, which is not nearly as strong in the amino acid. And you can do, again, ratios and differences, and you can see what sticks and what doesn't by ToF-SIMS. So this is an ongoing project. We're now sort of eagerly anticipating results on a whole variety of mineral surfaces and preparing essentially a library of mineral surfaces and to do this with ToF-SIMS. Our conclusions. First, I really want to just take-home message. Many, many different mineral surfaces. Any geochemical environment has the potential for chiral selection. And it selects these plausible prebiotic molecules. This may have been a contributing factor in creating local chiral microenvironments with concentrations of one chiral molecule or another. And I think the second idea is that using a microarray technology, merging ideas from geology and biology, bringing them together, we have the possibility of then approaching this in a combinatoric way where we can study potentially thousands of different mineral molecule pairs in a single experiment and therefore understand in much greater detail why certain molecules stick to certain minerals. So with that, I want to thank the Carnegie Institution, National Science Foundation, and especially NASA for funding in this research project, and I will really look forward to your questions.
Yes, sir.
So you've shown that chirality can be produced by reagents that select for some and not for others, but in extant life, it's produced by synthetic action. And so I was wondering how you reconcile the two and whether that implies something about the origin of life on Earth.
It's intriguing, yes, and life now basically uses pro-chiral reactants and creates chirally pure products, which is what the chemical engineers would love to do in every case, because it's a much simpler, more streamlined matter. Start with something simple, end up with a complex product in one step. That's a great way to do it. Life does that a lot. Life has lots of interesting enzymes and chemical reaction pathways that have evolved over time. To what extent is that pointing us to prebiotic sort of geochemical mechanisms? I'm not sure. I think that life keeps adding on complexity through selective processes, and natural selection drives to these more and more efficient mechanisms. In the beginning, it was not a selective process in the sense of competition. It just was some environments that concentrated this form and some environments concentrated that form. And it's from that milieu that you had to develop the first homochiral molecular systems. And what many people think is life arose from those systems. So there really is there's a difference between the competitive natural selective environment and just the selective environment that reflects local environment.
At one point you specified room temperature for seeing a chiral effect. Would you comment on the impacts of changing the temperature, either warmer or cooler, or having the mineral surface exposed to a temperature gradient?
Yes, there are situations where at cooler temperatures you get a much stronger selective effect, and indeed the way the chemical engineers study these effects on metal surfaces is they do it in a vacuum chamber where they can vary the temperature. What you will find is that as you raise the temperature, first of all, there are always a few molecules that are sticking there with just one point of interaction. And at say, 100 degrees centigrade or 150 degrees centigrade, they fly off. And you get up a little higher temperature. And there are always some molecules that are sticking on the surface with 2 points of interaction. So maybe you hit 200 or 225 degrees centigrade, and those molecules fly off. You hit 300 degrees centigrade before the things that are bonded with 3 points of interaction go flying off. And so this is a way of chiral purification in chemical engineering, is you basically bond a whole bunch of things to a surface, heat up the surface to the point where only the things, the three, that's the chiral interaction, remains. So temperature is very important, and the higher you go in temperature, you can certainly select things in a new way, but also you get stronger bonding at lower temperatures. So there's a trade-off. I think this is another case. I've made this comment repeatedly, like yesterday in the lecture, that cycling of conditions is critically important in the origin of life. If you're going to try to do chemical experiments that mimic origin processes, think always about how you can bring cycling into it, raising and lowering the temperatures, drying and wetting cycles, cycles of light and darkness. All these different cycles were very prevalent on the early Earth, and each cycle it winnows out the possibilities. In fact, you can think about each cycle as adding information to the system, and therefore making the system more selective. So I think that it's not only the temperature, but it's the cycling of temperature that can be really important in enhancing these effects. I've not used cycling in my experiments yet, but that certainly is a very interesting possibility.
Marcela.
Can chiral selection occur over the surface of ice crystals?
Oh, wow. Yes, ice does have chiral faces, absolutely. And I've not studied ice in detail, but certainly if you fracture ice, you're going to have a whole range of interesting chiral surfaces. And furthermore, I suspect that they will have both positive and negative charge centers. And furthermore, those charge centers, what is the HO distance in ice? Do you know? I know that it's a hydrogen bonded thing, so it's like there's two distances.
Yeah, 1 and 2.
1 and 2 angstroms. So you could have some very interesting geometries that match up with some molecules. So it's a very interesting possibility. And obviously I should have thought about that, but I haven't. But that's a very interesting possibility of ice.
Has anybody else thought of that, or can she do that for her dissertation?
Sounds great. Oh no, no one has discussed chiral surfaces of ices. Absolutely not. There's no literature there. That's a very, very interesting possibility. And another thing that's really interesting and no one has studied is the microbial interactions with these different chiral surfaces. And I'm not sure if there is any. I mean, microbes are much, much larger than the molecular scale features we're talking about, but it's conceivable that there is some different absorption. You may have, who's the fellow who is at Virginia Tech and then Maryland who did biological force microscopy, putting microbes on the end of an AFM and having them touch mineral surfaces to see whether they like to eat them or not, and measure the force. Well, you could do this on chiral surfaces as well. I mean, so there are a lot of intriguing ways that you could study these interactions, and I encourage you to think, out of the box on this stuff.
Yes, ma'am.
Yeah, I actually, this is more like the method in which you use to separate the enantiomers. How are you guaranteed, especially with the two methods that you described, that you do split them? I guess, especially thinking of medicinal uses or pharmaceutical uses, how are you guaranteed that I'm only getting the L or the D? I mean,
ask the FDA. I don't know. This is really an important question. Many drugs that we take, I mean, you and your family and your friends take. I would say the majority of the really expensive drugs that are out there have to be chirally pure. And how do they test it? How do they pure? I mean, you have to basically do quality controls with chiral columns. And there are ways of passing these molecules through columns and separating out the left and right hand and seeing if you get one peak or two. But it's not an easy job to test them and purify them. And I think when I buy drugs, I just sort of trust that they're okay. I also don't know of any case other than thalidomide that's so dramatic. So there are probably a lot of chirally pure drugs where there wouldn't be a huge difference if you took left and right-handed molecules.
Well, actually, as humans, we're really kind of based upon the left, right? So. Well,
okay. First, let me just. I'll come back to your question. The definition of left and right-handed is a completely arbitrary historical definition based on going clockwise around the carbon atom from the least to the greatest mass, and you go clockwise or counterclockwise, and that gives you left and right-handed. So the whole question of left and right-handed. Most of the amino acids in our body, according to that definition, are of the left-handed variety, although it turns out that in your gut there are microbes that coat themselves with right-handed amino acids so that they don't get eaten by your digestive system. But, so there's always some exceptions. But yeah, most of your body is left-handed amino acids. And by this definition, if you have left-handed amino acids, it turns out that the sugars are right-handed sugars. So in DNA and RNA and the other sugars you take. And there's now a dietary supplement that's available, which is left-handed sugar, which tastes like sugar, but turns out isn't digestible because it's the wrong-handedness. So I mean, there's all kinds of weirdnesses in terms of talking about left and right. Yeah, but yeah, your body is chirally pure in many respects. So.
You kind of answered it, but I was thinking in terms of especially medicinal testing, how are you going to know how your body reacts to it, whether it's right or left? I mean,
yeah.
I'm really skeptical.
I don't know. I know it. Well, it's a very important question. I guess use mice first. It's a very important question, and maybe we should all be a little more skeptical about. But that's why we have FDA and all sorts of drug testing procedures, and why they reject a lot of drugs that might otherwise be important. It's not my expertise, but it's a fascinating area.
I'd like to bring up an issue that was briefly mentioned last night, namely with the origin of life. So we get some separation on these different surfaces, but it's a relatively small scale kind of. You're not talking about this 100-kilometer area,
or even meters.
Yeah, right. I mean, it's
square millimeters or microns.
So, how does one get life going with such small regions of interest?
How else would you get it going, though? That's the real question. I mean, it's a juxtaposition of molecules in a microenvironment, and it's those molecules creating a self-replicating system that then takes over.
Yeah, but they
the
can't take
molecules
over
have
because
to be
there's
in
another
contact.
surface right next door that they're incompatible with.
And that's another experiment. And that's one of the amazing things about the early Earth. You see, they were just trillions upon trillions upon trillions of microenvironments that were chiral. Some of them weren't chiral. Different molecular selection on this face and on that face and in this pH range and in that salinity range. So you had all these different experiments going on. And in one of those microenvironments, the juxtaposition of molecules was such that a self-replicating system began spreading out. And once that self-replicating system got going, it ate everything else very quickly. So that's sort of a very simplistic idea. But I think it has to start out in a microenvironment because there has to be molecules interacting with molecules. It can't be a molecule over here and a molecule over here sort of thinking about each other. I mean, it really is a chemical process.
Right.
And so that's one of the reasons why the prebiotic soup is so problematic. A soup is too dilute. A soup, the chances of just the right molecules contacting each other in an aqueous solution, so you need to have a surface. And whether it's an oil slick, or whether it's a mineral surface, or whether it's an aerosol, or a dried up pond and a tidal flat, it's got to be a surface, because you have to concentrate your molecules and you have to bring them together in an environment. And if that microenvironment is chiral, all the better.
So is the idea that you start off with this speckled nature, so to speak, but that the facets that are similar eventually combine together into and take over. I mean, the chemistry that's been produced by those facets.
It isn't necessarily even that the chemistry of all the different facets combines and takes over. It's just that some place an efficient self-replicating molecular system begins. And when an efficient self-replicating molecular system begins, it's so much more efficient than anything else than it just...
But that could just be 100 square centimeters.
It could be...
what was the initial kernel?
A square micron. It could be a few thousand square nanometers.
That's what I'm getting at. I mean, you're talking about the origin of life as being in 100 square centimeters,
yes,
rather than a pond.
Yes. Yes. I mean, it's going to be in a pond or in a tidal flat or something, but it's going to, the actual origin of life is a group of molecules that starts doing.
You might call it the point.
And I so that it once it goes, and then it goes.
Okay.
And it may have happened 1,000 different places, in the sense that this locality, and these experiments started, and maybe they weren't very efficient and they failed, or the adjacent one ate it because it was just so much better. So I'm not opposed to multiple origins, but I think each of those origins is a local time and place, highly localized. And you're looking at microenvironments. That's what we're thinking about. Now, the other thing I have to say is that those microenvironments may have been influenced by many other environments, hydrothermal vents and comets and asteroids coming in, and Miller-Urey processes and different kinds of geochemical processes that will cause the juxtaposition of these molecules in that particular microenvironment. So it could be that all of those environments are important, but ultimately life began as a molecular process in a micro time and place.
In that same vein, so there seems to be a spatial bottleneck at some point. And I was wondering what, in the sequence of the origin of life, specifically, what was at what stage in the emergence of life is that bottleneck?
And that's the big question. In fact, I'm going to devote most of the next Origin of Life Gordon Conference to that. Is the question, is it metabolism first? That is, was that bottleneck, was that first thing just a group of molecules that created a metabolic cycle or network in that local environment? Or was it an information-rich molecule that somehow was first able to pass on information and make copies of itself? And that's the genetics first. So that's a big debate. There are many people who think it had to be that genetic molecule that somehow then used its environment to promote making copies, and some people think it had to be metabolism that just sort of mindlessly made copies of molecules without any sort of information context at all. And this debate is I think at this point unresolvable, but I think experiments, there are key experiments on both sides of the issue that might at least persuade people one side or another. And maybe somehow you had to have both. You had to, in one environment, you had an information-rich molecule that learned to make copies of itself, but really because it didn't have metabolism built into it, was very much limited in time and space. But then you had these self-replicating networks of molecules that kind of oozed around, but really were not able to do. And then they came together, and bingo, you had a merging of genetics and metabolism, and that's where things just took off. So we don't know. It's a very, I can wave my hands all day, but I don't have any idea at this point. It's just a lot of fun to think about.
Well, I think we better end here. Thank you very much for fascinating.